Is that better?
Yep, that's great.
Awesome. Okay. Right. Well, thank you, William. Thank you very much for the invitation. It is actually, I was looking at your website and noticed the illustrious list of speakers that you typically have at the Oxford Biological Society, and I'm honoured actually to be listed among them. It's slightly different, of course. I mean, I guess it gives you the opportunity now to have people from further afield that you otherwise never would have. I don't suppose that £5, whatever it is, a year membership, you're going to have the money to bring people in from afar, but nevertheless, I am happy to be here. As William said, so I work on plant hormones and plant development, and so my background, I guess, is molecular genetics. And when I was at Oxford, I was most interested in essentially the cell biology and genetics side of things, and I started specialising in plant genetics, I guess, when I did a project with Jane Langdale. And I just found, I remember doing a practical class on C4 and CAM photosynthesis, and I just thought photosynthesis was amazing, and I had to work on chloroplasts and genes and how things are made, and so that kind of fascination has just been maintained really until now. So it was 20 years ago I graduated, which is frightening when I think about it, but it's half a lifetime ago. But it's lasted with me. So I hope you guys are all appreciating what a great course you have at Oxford. I was just looking at the website. I think it's going from strength to strength. And now you've got this 4th year option as well. I think that's awesome. Okay, so listen, I'll move on. And as you can see in the background here, I've got a picture of beautiful, pretty flowers. These are, this is a picture I've lifted straight from Wikipedia. These are beautiful floral examples from southwest of WA. So right in the middle here, or lower middle, we've got the Anagoxanthus kangaroo paw. That's the emblem of floral emblem of WA. And I think you'll notice, those of you who are familiar with flowers, plenty of orchids, and there's a Banksia flower up here as well. So I just put this just to say that we are basically living, or I am living in what is known as a global biodiversity hotspot down here in southwest WA. Climate-wise, it's like Mediterranean, so it's very similar to Portugal. I don't know whether you still go on the Portugal field trip in the 2nd year, 3rd year. That's what the place reminds me of. There are a few various like sub-regions that are characterized by different types of vegetation. I'm just going to try and get my pointer up here. Laser pointer, there we go. Pointer, laser color green, much easier to see. Okay, and Perth is just on that little knobbly bit next to the 2 there, in case you're interested to know. All right, so we typically get dry, hot summers here. We're just coming out of summer, and that's associated with bushfires, as I'm sure you're well familiar with. But certainly before the white man turned up here in Australia, this corner of WA was full of a forest, essentially, especially the extreme southwest corner where you see eucalyptus forests like these guys. So these are karri trees. They're extremely tall, straight, beautiful timber. And these forests are like the Valley of the Giants, it's called down there. And they're really an impressive sight. So if you ever get a chance to come to WA, you must go to the karri forests. Here's kangaroo paw again. Here's a Banksia inflorescens, not a flower. I have to be careful with my language. Animals, of course, there's lots of endemic beautiful animals here too. We've got the honey possum sitting on a Banksia inflorescens. Marsupial, of course. And one of our more elusive customers, he's a, oh, goodness me, I've already forgotten, but we've got to look here. Bilby, thank you. Bilby, he's nocturnal, big ears, I don't know, helps him hear his food, something like that. And my personal favorite is the red-tailed black cockatoo. Okay, these are increasingly, sadly, quite rare now, especially in the city. There are a few pockets here and there, but they're certainly a threatened species. And in case I need to tell you any more, I don't have any pictures, but we do have the best beaches in the world. Beautiful white sand, especially along that south coast. I also thought I'd just say a little bit about UWA, the University of Western Australia. Certainly when I was at Oxford, you know, as far as I was concerned, the only universities were those in England, or well, UK, I suppose. But I I came across UWA when we had quite an active contingent of people who would come from UWA to study in the Department of Plant Sciences at Oxford, actually. And so there was a regular, every one or two people coming for fellowships, research visits, that sort of thing. And so word got around about some of the plant sciences getting done at UWA. And if you look carefully here, so this is obviously a shot. Here in the background is Kings Park, one of the largest city parks in the world. It's basically bushland with a botanic garden at one end. The city of Perth, as much as it's a city, there's a few skyscrapers in the distance. This is the Swan River, and this is the main Crawley campus of UWA. And my office, where I'm talking to you from now, is in this grey building, the Baylis building. Okay. So, all right, we're not in the top 10 like Oxford, but we're very proud of the fact that in some rankings we are, it's certainly in the top 100, and it's not to be sniffed at. So we are a research-intensive university, and biological sciences and agricultural sciences, we are among the best in the country, definitely. The School of Molecular Sciences, people ask, well, what's molecular sciences? We came up with that name because traditionally this department was chemistry and biochemistry, and the best name that we could think of without offending anybody was something generic like molecular sciences. So there's 40 odd academic staff plus postdocs, research staff, postgraduate students, PhD students doing most of the research, and then within that we teach genetics, biochemistry, and chemistry undergraduate degrees as well. Anyway, you don't need to know that, you're already in the best biology degree course in the world. So biology, yeah, okay. So what I'm going to talk about is primarily a research talk, I suppose. I've tried to make it as accessible as possible, but there will be some data. Okay, there's going to be some graphs. So hopefully it's going to stretch your mental capacity a little bit as you follow it. But by all means, write down questions, anything you don't follow, or something that occurs to you along the way. Please, interrupting's a bit hard, I suppose, in this format, but save it up to the end, and I'll be happy to answer anything that occurs to you. And so anyway, right. So I guess plant development is my thing, and what I like about development is that it's a progression of decisions, I suppose. And this is unique, by no means unique to plants. Animal development is much the same. It's all about cells interpreting their positions relative to other cells and their environment, and making decisions as to what they're going to become next. And in so doing, you end up with tissue types and therefore organs, and hopefully a functioning organism. But one of the impressive things about plants is that most of its development is post-embryonic. So the embryo is sitting there, at least in flowering plants or seed plants, it's sitting there in the seed in a form of stasis until germination takes place. And then it's got to make all its extra embryonic organs, all its root system, all its leaves, ultimately its flowers and that sort of thing. And so plants are very plastic. Their phenotype very much depends on the environment that they're growing in. And so there's a lot of interpretation that needs to happen during development to make these, I suppose, a successful life cycle. So an example of critical decisions for if you're a plant would be something like, should I germinate now or later? Should I flower now or later? And when should I grow very tall to escape competition, or should I try and grow compact so that I don't fall over in the wind? All these sorts of things are decisions that plants have to make. And the thing about a lot of these decisions is that they're typically irreversible. So there's a strong selection pressure to make decisions that are robust and also correctly timed. Now, making a developmental decision, I guess, is informed very much by not only internal cues like the clock, so time of day and at what stage of development you are, but also external cues, and they can be things like temperature and light, nutrients, and as you'll soon see, chemicals as well. And one of those, as I said, seed germination is a major decision to make. If you think about the seed, it really is quite a marvel of evolution, because it allows a prolonged cessation of the growth cycle for long enough for preferred, I suppose, optimal growth conditions to come around. So it was a key innovation, and I suppose the major downside of it is that if you've made that decision to germinate, it's pretty much it's make or break it, right? The seedling either establishes itself and grows up to make flowers and therefore reproduce, or it fails pretty early. So getting that timing right is really crucial. And so a lot of seeds in many species are subject to a process or a phenomenon known as seed dormancy. And we know what dormancy means, okay? So buds are dormant, things are asleep, essentially. And to make it straightforward, a seed is considered to be dormant if it fails to germinate under conditions that otherwise it normally would germinate. So that's a bit kind of confusing. So, you know, how can a seed not germinate when it should be germinating? Well, it's because a number of things have to be met perhaps at certain times or in a certain order in order for dormancy to be broken. And those things or events are presumably correlated or related to what the seed considers to be ultimately optimal conditions. So factors that would regulate seed dormancy and therefore the decision to germinate or not include things like local nutrients in the soil. So nitrate can stimulate germination and break dormancy. Hydration, of course, and seeds in the soil, they'll go through cycles of being dry and then wet and then dry and then wet. And this dry-wet cycling can affect dormancy. So sometimes a seed has to go through 1 or 2 cycles before it's actually competent to germinate. Temperature, very important. Obviously, a seed germinating when the weather is too cold is not going to have a good chance of surviving. So it's good to germinate when things warm up again in the springtime. And hormones, internal hormones, especially abscisic acid, are very important. So abscisic acid inhibits germination, gibberellic acid promotes it. And so things that can regulate the expression of genes that are involved in the synthesis or breakdown of those 2 hormones can ultimately lead to germination. All right, so as William mentioned at the beginning, so I have been working on these things called Karrikins, which are abiotic chemicals. So that means, you know, they're not made as part of a biosynthetic process, but instead they simply result from partial combustion of vegetation. So it's sufficient basically to burn cellulose or even xylose, a simple sugar. And if you only burn it under limiting oxygen conditions, you get partial combustion products, some of which are these things called Karrikins. And there's some structures here on the right. They've all got this bicyclic structure. So we've got a 6-membered ring and a 5-membered ring. So this 5-membered ring is called a butenolide group, and I'm going to use that term butenolide a fair few times in this talk. So this is the conserved, and this is actually the bit of the molecule that's important for its activity. But before we get there, I'll just say what its activity really is. And that is that they can promote the germination of seeds that otherwise would not germinate very quickly. So this is a picture of Arabidopsis seeds. They're freshly harvested, and when seeds are fresh, they tend to be dormant. They don't germinate immediately. And so if you sow them just basically on water, so this is agar dissolved in water, you only get very limited germination. So we've got a couple here that have woken up. But if we add just 1 micromolar of carb-1, so this compound here, this is the most abundant compound found in smoke, then you obviously get quite an appreciable number germinating at the same point in time. So this could be interpreted as meaning that Karrikins are breaking the dormancy of these seeds and promoting their germination. And that's why we'd call them a germination stimulant. And in real life, a good example of how and when this happens is here in Kings Park. So locally, you remember that picture I showed you earlier? In 2009, there was what turned out to be a deliberately lit fire in Kings Park that, I forget the statistics, but it burnt through a few hectares of bushland. And so that was in the sort of summer of 2009. And of course, 2010, so about March time, about this time of year, that was when the weather had cooled down a bit and seeds then started to germinate. And this species called yellow tail flower, or Anthocercis litorea, had not been seen in Kings Park for 20 years, basically since the previous fire. And this is a classic, what we call a classic fire follower, in that it's basically requires a fire in order to germinate. And the ecological reason for that is quite clear, of course, because a fire clears vegetation, allows more light to reach the ground, also releases nutrients into the soil and that sort of thing. So it's advantageous for a plant to recognize when a fire has come through and sprout up like this. So this was a very visible manifestation of this phenomenon, and it happened just as I turned up to start working on these Karrikin things, and somebody said, hey, come over to Kings Park and check these plants out. So there we go, it's real, it does happen. Okay, so yeah, there is a little bit of chemistry in this, but nothing too frightening. Okay, we'll keep it simple. So I mentioned those Karrikins and how they are butanolides, and I've highlighted the butanolide ring of Karrikin 1 here in red. All right. Now, when these compounds were isolated originally, so this was 2004, Gavin Flamati here at UWA had been searching for the compounds in smoke that stimulate seed germination. So he did that by separating smoke water using essentially HPLC-based methods, so the chemical separation methods, into simpler fractions, and then testing those fractions for their ability to germinate seeds. And so eventually he found Karrikin 1. That's the short version of that story. And they immediately noticed that if you look at other compounds found in the natural world, especially plants, that this butanolide ring had been noticed before in this other class of compounds called Strigalactones. Okay, they're called lactones because this butanolide is a form of lactone. That's a chemical moiety, a chemical structure. And strigo because they were identified as compounds that a parasitic plant called Striga responds to in the soil. So just back up a little bit. Parasitic plants, or especially root parasitic plants, essentially the seeds lie dormant in the soil, and when they detect a nearby host, something like, and typically these are crop plants, so maize, millet, sorghum, tomato, the parasitic seed will germinate, produce this thing called a hostorium, and latch onto the root of the host and then start parasitizing it. Now it turns out that how does the parasite recognize a host is nearby? Well, it's detecting these compounds called Strigalactones that are released by the root of the host. So these compounds were identified in 1966, so a long time ago. They've been known for a long time, but besides how they, that they were actual stimulants, no one knew how they were made, how they were detected, how much there was, what diversity there was. And it turns out there's at least 20 different Strigalactone compounds that all look a bit different in this black region, but they've all got this butanolide ring here in red. Okay. Now, the obvious question was, or one question that came out was, why would a plant like millet or maize release compounds into the soil that would attract a parasite? That is not a good thing to do from the point of view of the host plant. And the answer is, of course, that these Strigalactones are doing other things. Actually, one of the main roles of Strigalactones is to attract arbuscular mycorrhizal fungi. So these are symbiotic fungi that live in the soil and form associations with plant roots to help gather nutrient uptake from the surrounding soil, especially phosphorus. So under phosphorus limited conditions, so in poor soils, plants will often release more Strigalactones into the soil to encourage arbuscular mycorrhizal fungi to come in and associate with the roots. And we'll see some pictures of those in a short while. All right, so, okay, plants are releasing Strigalactones to attract fungi. The other thing that these things are doing, these Strigalactones, is they have a hormonal function within the plant, so they regulate plant development. And there's a lot of things that they're involved in now that's been discovered since 2008 when this was confirmed. And I'm not going to talk about this very much, except to say one thing, that the most obvious thing that Strigalactones do is they inhibit the outgrowth of buds in the shoot. So when a leaf is made by a plant, certainly an angiosperm, there is an axillary bud made in the sort of crevice between the main stem and the leaf itself. Right, so this bud here can grow out to form a branch. That's how basically shoots branch. But there is tight control over when and whether those buds will activate and form a branch. And part of that control involves these strigalactones. So strigalactones are made in various tissues in the plant, and they effectively inhibit the outgrowth of buds. And as a result, we know that because mutants that cannot make strigalactones have buds that grow out prematurely. And we'll see a picture of that as well. They tend to look bushy. Okay, so strigalactones and karrikins both have this butanoid moiety, and they are related, and that will also become, why I'm introducing this will become clear in a moment as well. Okay, so they're both germination stimulants in a sense, but actually they are different, okay? And one way that we know that they're different is just through the fact that not all seeds will respond to both compounds. So on this graph, we're looking at a germination graph essentially here. And one species in purple here is Brassica tournefortii. This is a weed. We will also call it wild turnip, and it responds very nicely to karrikins. So on the x-axis, we've got concentration in molar of karrikins. So here, -9, that's on nanomolar, 10 nanomolar, 100 nanomolar, -6 is a micromolar of Car1. So we're getting germination stimulation even at a nanomolar. But this species, Brassica tournefortii, does not respond very strongly to this strigalactone compound. Right? So even though it's really sensitive to karrikins, it's effectively blind to strigalactones until you hit, until you get up to 10 micromolar, and that's high. When we're talking about hormones, 10 micromolar is a lot. If we look at another species, this is a parasitic weed, which of course has evolved to detect strigalactones exuded by host roots. This one is very sensitive to strigalactone at nanomolar, but is completely blind to karrikins. So if these two compounds, strigalactones and karrikins, were the same thing and were working in exactly the same way, then we wouldn't expect this distinction between different species. So that's one of the clearest bits of evidence that they're different. And of course, we now have better evidence that relies on genetics. And I will come to that in just a moment. So that introduction maybe was longer than I thought it was going to be, but anyway, what I'm going to show you now is 3 short stories that I think illustrate some of the stuff we've done over recent years. The first one is how we discovered the karrikin receptor protein. The second one is about how karrikin perception can actually help weeds become invasive. And the third one is what I hinted at in my little abstract was, do butanolides have a role in organisms other than plants? Okay. All right, so move on. All right, so one of the ways if you want to understand the molecular basis for some kind of process, especially a developmental process, a mutant screen is a very powerful way of doing that. And when we work with, so plant geneticists often will work with a species called Arabidopsis Thaliana. It's a brassica, and it produces a lot of seeds, and it was the first plant to have its genome sequenced, all that sort of thing. So it's a toolbox for doing genetics in plants. And you can generate mutants by mutagenizing them with something called EMS, a mutagen, chemical mutagen, or X-rays. And then in this case, we did a screen where we sowed the seed on media that contained karrikins, Car1, and we were looking for seeds that failed to respond to that karrikin. So this was started by Dave Nelson, who now works at University of California, Riverside. And he undertook this screen, which was quite brave, I think, because seeds are a little bit, well, they're a bit awkward. Sometimes they can refuse to germinate for lots of reasons. But anyway, he managed to isolate a mutant that failed to germinate on karrikins. And what he did was he was taking these seeds off the karrikins and putting them onto nitrate medium, which would then break the dormancy. and then get the seed to germinate. And then you could recover the seed from the next generation and screen them again on Karrikins. And if they still fail to germinate in response to Karrikins, that's a good sign that you've isolated a mutant that is now insensitive to Karrikins. So remember, if we want to understand what genes are required for plants to perceive and respond to Karrikins, hopefully one class of mutants we could look for are those that fail to see it, essentially. So we were expecting to see genes required for this response, and we did so. So we found Chi1, which I won't tell you about right now, basically because Dave did it, but also, and then I found Chi2. Okay, so when people name genes from mutant screens, they will give them a name based on the phenotype. So Karrikin insensitive. Karrikin insensitive 1 was the first gene. It's got another name called MAX2. And then Karrikin insensitive 2 was the second gene that we found. And this is here just a little illustration of what the gene looks like. So I've just sort of colored the various bits of the locus in different colors. So this thin black bar is the intron that doesn't code for any proteins. And then there's 2 exons that do code for proteins, and we isolated 2 mutant alleles. Okay, it's not too important what they are, but they both have the same phenotype, and the phenotype is, as you would expect, they don't germinate in response to Karrikins, but they also have this very familiar long hypocotyl phenotype. So a seedling, when it germinates, produces, or at least this type of seedling, when it germinates, produces a kind of embryonic stem called the hypocotyl, upon which the cotyledons are on top, and then the leaves will form after the cotyledons. And this row here shows the wild type, the normal Arabidopsis seedling phenotype. And so when you grow them under certain light conditions, you get a seedling that's, 6 or 7 millimeters tall. And if you grow them on Karrikins, that hypocotyl gets shorter, okay? And if you grow them on Karrikin 2, it gets even shorter. So this species, Arabidopsis, is more sensitive or more responsive to Karrikin 2 than it is to Karrikin 1. But this mutant, Chi2, this particular allele, Chi2-2, they're both the same, it fails to show this reduced hypocotyl length phenotype. Right, so this is another example of it's insensitive because its hypocotyl cannot respond to the Karrikins that are in the medium. So that gives us 2 nice phenotypes, and and actually it's easier to measure seedlings than count germination. It doesn't sound that that's the case, but seedlings are they behave much more consistently than seeds do. So often we rely on the seedling phenotype to tell us what's going on. Anyway, to sort of cut a very long story short, we cloned the gene. We actually crystallized the protein. We solved its structure using X-ray crystallography. So that was published in 2013. And this is its structure. It's a globular protein. It's quite small, about 30 kilodaltons, and it's got 2 main domains. And it's got this green core domain, so 7 alpha helices, 1, 2, 3, 4, 5, 6, and maybe there's one around the back, and a few beta sheets. And then on top of that is this purple cap domain of 4 alpha helices. And if you turn the protein around a bit, you can see it a bit more clearly here. It makes 2 V shapes, one on top of the other. And what this actually forms is a channel through which the Karrikin, or we'll call it a Karrikin for now, can go and enter into the active site of the protein, which is here in yellow. So in the middle, we've got this 3 amino acids, serine, histidine, aspartic acid, that form an active site. And this type of protein is classified as an alpha-beta fold hydrolase. So the alpha-beta fold is this shape, basically, of the alpha helices and the beta sheets. And it's a hydrolase because it uses water to break things apart by hydrolysis, and it's catalyzed by these 3 amino acids at the center. Okay, so that's what it was kind of characterized as bioinformatically. Obviously, we had to do a fair bit of work to show how it worked enzymatically and how it works as a receptor, and that's still going on. And I might show you a little bit more about that in a moment. But what was immediately striking was that actually Chi2 is a member of the same family as another protein called D14 or dwarf 14. And dwarf 14 is a strigalactone receptor. Okay, so let me just briefly show you what a strigalactone receptor mutant looks like. These are rice plants, and this mutant was first described in 2009, and it was called dwarf 14 because it's short, right? It's a short plant. In fact, this is D10, dwarf 10. And here's D14, and it's clearly shorter than the wild type. And if you, D10 is a mutant that fails to make strigalactones. D14 is a mutant that fails to perceive strigalactones. And you can make a double mutant and they look, the double mutant looks the same, which suggests that these two mutations are affecting the same developmental process in essence. So one of the things I did when I found Chi2 was I also looked for the D14 homologue, the equivalent D14 gene in Arabidopsis. And Arabidopsis is a lot easier to work with than rice because it grows much quicker and it doesn't need lots of high light. And what you can do is you can, one of the great things about Arabidopsis is you can go to public databases, collections, seed collections, and order mutants in a gene of your choice. And we could order a mutant in the D14 gene, and then we looked at its phenotype, we grew it up. And what the arrows here point to are the extra branches. Okay, this plant is shorter, it's dwarfed, like in rice, and it's got more branches, those axillary buds. Remember I told you strigalactones inhibit the outgrowth of axillary buds? Well, they grow much more strongly and profusely in the D14 mutant. Okay, so we'd isolated a mutant in Arabidopsis, and as I said, Chi2 and D14, they belong to the same protein family. Now, if we look at this little diagram, we basically have two groups of proteins in plants. The first group is the Chi2 family. It's got members in dicots, monocots, gymnosperms, lycophyte ferns, mosses, bryophytes, right? So all of, and algae as well. So all of these are ancient plants. So Chi2 proteins are very ancient. All plants have a Chi2 protein, at least one, sometimes many more. The other group of proteins in this family is called the D14 group. And these are unique to seed plants, right? So the lower plants, basal plants like Marcantia, don't have D14 proteins. And so what we think happened was early on in land plant evolution, there was a gene duplication event prior to the emergence of seed plants that allowed a D14 to evolve. Okay, so essentially D14 and Chi2 are homologues in the same protein family. So they've got very similar structure and very similar function. And here they are. Here's the structures of both those proteins. This is Chi2 I just showed you, and here's D14. And you can see that if you can mentally overlap them, they are effectively the same shape. All right. But there are some differences, and those differences are crucial because D14 is a receptor for strigalactones, and Chi2 is a receptor for Karrikins. Remember, Karrikins and strigalactones both have that butynylide group, so they have got similar, both these proteins that are from the same family are detecting similar chemical compounds. All right, so I think when I'm just going to finish this little bit of the story with this point, and this is why I think it's exciting. Basically, Karrikins are sort of, you know, they're interesting, but you only normally find them from fire. So I think the key point here is that Chi2 is not there to perceive fire, it's there to perceive something else that's really important for plant development. And I think that's a new hormone that we don't know about. And the reason for this basically is, as I just said, Chi2 is present in all plants, not just those that see fire. Okay, so it's not just in fire zones, everyone's got them. Plants that don't have Chi2 function have got a number of problems. One of them is their leaves are all misshapen. Here's Chi2 mutant, it looks pretty sick. Another one is that they lose water at a greater rate from the leaves. So this red line shows that the water loss in the mutant is much faster than water loss in the wild type. So they've got a problem with regulating their stomata. They've also got a problem with growing under the inappropriate to the light conditions. So very briefly, wild type plants, if you expose them to some far red light, they think they're growing in the shade and they grow taller. Cockatoo plants already think they're growing in the shade before they've even seen any. So these seedlings here are just grown under white light. These seedlings are grown under simulated shade, and they basically look the same. So they can't tell the difference. They don't know when they're growing in shade. And that's a pretty major problem, because that's how plants escape competition from neighboring plants. And finally, we think Cockatoo is really important in these arbuscular mycorrhizal fungi symbioses. They are, here's a mutant in a grass called Brachypodium, a Cockatoo mutant. And these little dark patches inside the root are the arbuscules where the fungi have invaded the root and formed these exchange structures, and they've been stained here with some sort of dye, and the mutant cannot make them at all. All you get are these occasional fungal hyphae that are wandering around the root, bored, wondering what to do. Okay, so we think clearly Cockatoo is very important for lots of things in plant development, and what we don't know is what it's perceiving. We know D14 is a hormone receptor for Strigalactones. We know what the compound is. Cockatoo, we don't know what the compound is, and that's what is, I suppose, my mission at the moment, and it's a tough one. It's a tough one. Now, look, I'm well aware of the time, so what I think I'm going to do is skip the second story and just move on to the last one, because I think that's where I think the future is, and hopefully it's going to help us solve this problem of what Cockatoo is really doing in plants. So if you'll just bear with me while I find the right slide. Okay, so question 3. Do butanoids serve as signals in other species except plants, other than plants? I wouldn't be saying that if I didn't think they did. Okay, so this diagram again, I didn't mention this the first time, but yes, we've got all of these homologs in plants, Cockatoo and D14, but there is also a very close homolog in the bacterium Bacillus subtilis. And I've used Bacillus here as an example to represent lots of bacteria. So it's not just Bacillus that has this protein, but they are found in very diverse groups, so proteobacteria, firmicutes, and both gram-positive and gram-negative bacteria have this protein called RSBQ. It's by no means ubiquitous, so not all bacteria have it. E. coli doesn't have it, for example, but Bacillus and its relatives all have it. And here's a picture of Bacillus, false-colored electron micrograph. And RSBQ, so this is the homolog of Cockatoo, is necessary for the bacterium to respond to energy stress. So if you imagine how a bacterial culture grows, all right? So you put bacteria in a nice, fresh, liquid medium, they get very excited by the fact that they've got all of this yummy food to eat, and they grow exponentially after a lag phase until they reach a natural plateau when they start to run out of food, okay? And this is the stationary phase, and what's happening here is essentially they're running out of energy. And this is a stressful situation. It requires a very different kind of metabolism compared to exponential growth when they're just multiplying as fast as possible. Okay, now they've got to deal with the fact that they are under nutrient limitation. And so this means we have a change in metabolism, a change in gene expression to account for this new growth environment. All right, so I'm telling you that because RSBQ is a protein that ultimately regulates the expression of genes that are used to cope with this transition into stationary phase. All right. So mutants that don't have RSBQ, they do not properly sense the fact that they're running out of energy. And as a result, they don't activate the genes that are required for stationary phase growth. And so they tend to keel over and die quicker. Basically, they are not competitive because they can't cope with the changing growth conditions. All right, so that's RSBQ, and that's quite old. I haven't done any of that work myself, but what we've come up with is we've made the link basically, and we said, okay, we know that these proteins here in plants are receptors for butanolide compounds. They do signaling. What is RSBQ perceiving? And the truth is, nobody knows, right? People know what it does. They know what the phenotype of a bacterium that doesn't have RSBQ is, okay? But they still don't know how it actually works. And we think that it's actually a butylenide receptor. And we suppose that RSBQ is perceiving a butylenide compound that's made when bacteria undergo energy stress. And I'm just going to show you two bits of evidence that suggest, well, explain why we think that is. The first is what we're working with here is purified protein. So we make purified Chi2 from plants or purified RSBQ from bacillus, and we basically make it in E. coli and purify it using chromatography, and then we can assay its function in a test tube, basically. So what we're showing you here is a melt curve of a protein. And what you can do, so proteins, of course, they denature when they get warm, and they open up, and they will show a maximum point of melting, which is called melting point. Okay, and this is all revealed using a fluorescent dye. I won't go into how it works. But what you do is you can take the protein and you can mix it with increasing concentrations of a compound that you think interacts with that protein, and you can look to see whether it changes the melting temperature, because that can be used to infer that it's changed the shape of the protein. So if the ligand, this compound, and the protein interact and they change the shape of the protein, we expect it to result in a change in the melting profile of the protein. All right, so we're going to show you some rainbow graphs here where we're increasing the concentration of this butylenide compound here called desmethyl-GR24, but don't worry about that. So at 0, we have a melting point of around 45 degrees, something like that. And as we add 10 micromolar, we start to see a decrease in this peak here. And at 25, that peak's completely gone. And it's starting to get replaced with this second peak that is at a much lower melting temperature, something more like 35. Okay, so what we've done here is by adding more and more ligand, we've progressively affected or changed the conformational shape of the protein that was in our test tube, and as a result of that, we infer that this ligand has interacted with the Chi2 protein. Now, it doesn't do this with every ligand. Okay, we can change the structure of this, and we don't get this response. But the key point here is that we basically get the same thing if we use RSBQ from bacteria. We've got a melting point at a higher temperature, we add the ligand, and we get a second melting point at a much lower temperature, which suggests that the protein has been destabilized by the ligand. It's become more prone to melting. And actually, we get this is really quite nice because you have two very clear peaks without and with the ligand. So to us, we were very excited when we saw that, because that's a nice little assay that shows actually for the first time what this protein might do functionally. The other thing is we've got some in vivo evidence, right? So where we actually look at what happens to the bacteria themselves when we add these kind of compounds. And the way this works is very, very briefly, as I said, RSBQ activates this transcription factor, which can activate downstream genes. And we can measure the expression of downstream genes subsequent to the RSBQ being activated using a reporter gene. In this case, it's LAC-Z, which makes beta-galactosidase, which is an enzyme that we can measure the activity of in the lab. And all I want to show here is that as we increase the concentration of this DGR24 compound, we actually decrease the activity of this reporter gene. And we decrease it towards the level that you see in an RSBQ mutant. All right, so this is the mental gymnastics we have to think about here. This RSBQ mutant, if we don't have it there, this LAC-Z gene never gets activated, or only gets activated at very low levels. We can phenocopy, we can regenerate that response by exposing the bacteria to increasing amounts of desmethyl-GR24. And as we get to 10 micromolar, this expression has basically been suppressed. Now, this is exactly the opposite to what we were expecting. We thought we would add GR24 and the levels of expression would go up. But in fact, they went down. And so at first we thought, well, this is clearly wrong. But actually, we thought about it a bit more. And this is consistent with the idea that this compound is interacting with the RSBQ protein, but rather than activating it, it's actually poisoning it. It's killing the protein. We still think this, so it's probably, this compound is not able to turn RSBQ on, but it can turn it off, which is nevertheless good evidence that it can at least respond to these butanoids, even if we haven't got quite the right one for turning it on. So then the question is, well, can we find the compound that bacteria make naturally that activates RSBQ? And the method we have, this is synthetic biology to some degree, but it's very similar to that previous reporter, but instead of LAC-Z, instead we've got this bunch of genes, which are operon, thank you, should know that word well. And we've got an operon that encodes the, gives the bacteria the ability to make luminescence. Okay, so they glow in the dark, literally. They make bioluminescence in response to the activation of this upstream promoter. So RSBQ, if it gets activated by some interesting compound, will activate sigma B, which will sit on this promoter and then activate downstream expression of the luminescence operon, and the bacteria will glow. And we can measure that glowing using a microplate reader in the lab. And what we would expect to see is a compound that activates RSBQ will gradually increase the luminescence signal, whereas an inactive compound would have next to no effect. So the way we went about this, I've got a student in my lab, Kim Melville. What he did was he took this bacterial strain and he exposed it to chemical extracts from stressed bacillus cultures. So he grew cultures of bacillus bacteria in a big flask until they reached the stationary phase and therefore started running out of energy. They got stressed. We hypothesized that they would make the special compound that activates RSBQ. He then harvested those cells, so he centrifuged them, he dried them down, he extracted using solvents the chemicals present in the pellet of bacteria, and then separated them very basically using methanol water solvent extract separation techniques. And he then applied those extracts to the bacteria that were expressing this luminescence reporter gene. And he was looking for strains that would, he was looking for fractions that would cause an increase in luminescence. And I won't go into the details of what these various lines are, except to say that this blue line where he's fed it with basically nothing has stayed relatively constant. And then this yellow line, which is one of several, which I'm not showing here, and the gray line are showing a spike in luminescence. And that happens within about half an hour, 20 minutes of applying the compounds to the bacteria. And it peaks at about an hour and then it starts to drop off again. Okay, so our mission now basically is to separate these chemical compounds that we think are responsible for this increase in luminescence, because we believe somewhere in that mixture there will be the compound that activates RSBQ. And finally, what we hope is that we'll also find that compound in plants. So that's the big plan. I've said it publicly now on Facebook. I don't know whether all my competitors will find it, but that's what we're doing, okay? So we'll see. All right, I had better wrap up, and I will just briefly summarize what I've said. Karrikins, they're abiotic, they're not made by organisms, but they are germination stimulants that actually were they were a bit of a red herring, but they led us to discover something new about plants that we'd never known before, some new signaling pathway that relies on Cockatoo to regulate plant development. Karrikins and Strigalactones, we know they're both bioactive butanolide compounds, but nevertheless, we're still looking for the compound that plants make that presumably activates Cockatoo. It's an assumption, but there's good evidence for it. I should probably say why we're even interested in doing this in the first place. Well, of course, if we can regulate all of those things like drought tolerance, leaf development, seed germination, shade responses, these sorts of things, we need to know how the system works so we can effectively control or modulate how plants grow. And of course, plants are food. That's why plants are really important. And therefore, we need to make more food on less land with lower CO2 emissions, all the rest of it. We need new ways of boosting crop performance. So that's why we need to be interested in hormones. All I'm sorry I left that right to the end. Anyway, so, and finally, as I just said, we think butanoids are more than simply plant signaling compounds. In fact, I would not be surprised if plants and bacteria are signaling to one another using this sort of system. So that's another exciting possibility that we're going to look into. All right, I just need to say thank you to all the people who've done this work. I've got various colleagues here at UWA, chemists, Gavin and Adrian, Georg, he's a synthetic biologist, Charlie, a structural biologist, for helping me solve these protein structures, lab members, past and present. So this was a relatively recent photo. Frank now, unfortunately, has gone back to China. Hugh here is a PhD student who's stuck in China because he went back for Chinese New Year in whenever it was, early 2020, and is still there, as we know why. And then I've got some buddies around the world who help me and share beers with me whenever I get to see them. Okay. Oh yes, and money, of course. Couldn't do it without the Australian government, UWA. And Plant Energy Biology is this group of scientists who like to exchange with Department of Plant Sciences in Oxford. Okay. Anyway, so that's me. That's the story. I'm sorry, I probably haven't left much time for questions.
Right. Thanks very much.
Completely underestimated how long it would take.
Thanks very much for your talk, Mark. So we do have some time for questions, if a few minutes at least. If people would like to unmute yourself and ask questions or put them in the chat, or if you're watching the Facebook live stream, we still have about a dozen people on there. If you put questions in the comments, then I can ask Mark. But in the meantime, I was curious about whether you've done any work in any of the early diverging plant lineages. So whether Cockatoo has been looked at in bryophytes or perhaps in green algae or anything like that.
Right. Yeah. Yes. So, so in one paper, what I did is I cloned genes from Marcantia and Selaginella, and tried expressing them in bacteria and in Arabidopsis. And basically, you can take a Cockatoo homologue from Selaginella, and it will complement the Arabidopsis mutant phenotype. But the interesting thing is that it doesn't respond to Karrikins. And instead, we have very recently published a paper together with Junko Koizuka, who I didn't put on the slide there, very naughty me, who has been working on Marcantia and the function of these proteins in that species. And so we found now that butanolide compound has got a methyl group off it, but if you take that methyl group off, it is now much more active in species including Marcantia. So the system appears to be the same. Mosses, oh my, they've got like 13 of these genes, and there's this enormous paper that is under review. I reviewed it. Oh, maybe they don't know that. And so it presumably will come out, but it's a very complicated story. But it looks like mosses can respond. They make strigalactones, and they also make or have this Cockatoo signaling pathway as well. It's complicated, but yes, early plants are a very informative system to use for understanding this.
What about green algal systems? Is that something that you can, because something like Chlamydomonas is quite a distant group that is perhaps tractable?
Yeah, so I don't, oh, now you're asking. I don't think Chlammy has a Cockatoo. So I put Nitella on there. So the Charophyte algae, the sister group, they definitely do have Cockatoos.
Yeah.
And people have done things like treated probably Cara itself actually with these strigalactone analogs like GR24 and observed a change in the I don't know, the length of the internodes or whatever it's called in algae. Unicellular algae pass. So it doesn't look like they've got this system, so they may well have lost it. Because it's a big mystery to me how we've got a group of proteins in plants and a group of proteins in bacteria, and it's not obvious where the homology there is. So was there horizontal gene transfer at some point? Did they truly all evolve from a single common ancestor? I don't know. Cyanobacteria, so on one thought, I thought, oh, okay, maybe it came via the chloroplast. But Seneca cystis and cyanobacteria don't seem to have them as a general rule. There might be the odd one that does. So it's a mystery where they've come from. So where in plant evolution they first emerged, I don't know, but it seems that only the most closest sister group to the land plants have got this signal pathway.
But it is in Bacillus, which is, of course,
But it
a
is in
long way
Bacillus.
off.
It's a very long way off, and I still, I'd love to know. I think the trouble is it's so far away and they're so divergent that actually you can't work it out with homology and phylogeny building.
And I suppose with bacteria, horizontal transfers are,
yeah,
of course, quite
it's
likely.
a mess. It's a complete mess. So working out the ancestry there is going to be hard. We're going to try and do it functionally, actually. So we think that if we sample RSBQs from different bacteria and see whether they have the same function as Bacillus, that will at least tell us, has that core function been retained, even though they're probably regulating different things. So one thing's regulating this sigma B transcription factor, but in other proteins, in other species, it's regulating things like biofilm formation and maybe secondary metabolite production, all sorts of things. So there's a lot to the. So the one thing about academic science is you have to stay focused, because you can quickly get swamped by question and idea, and then you go nowhere. And so there's a fine balance between sort of pushing new ideas and just getting stuff done.
So Maria has asked in the chat about a recording, and I can send the recording around in the newsletter, if that's all right with you, Mark, in the email list.
Absolutely, yeah. No, I'd like to, because apart from anything else, I keep a note of these things that I do, and the more people who see it, the better, as far as I'm concerned.
Recruit people into the field. Is that your intention?
Well, yeah, and actually, of course, I missed the punchline at the end of my talk there. I was in a hurry to finish, but if anybody does ever fancy coming over to Western Australia to, I don't know, do a PhD, you're always welcome. Oxford graduates, always welcome here. You have a very high chance of getting a scholarship. That's all I can say, because we think you guys walk on gold or something. I don't know. Yeah. So, but I mean, even if you just come to a holiday, I thoroughly recommend it. Right? I mean, people often come to Australia and they bypass the West, and I think they're really missing something. It's a stunning part of the world, and great place to live.
And that's after the COVID borders have reopened.
That's right. Yes, I mean Australia is many things, but it's also very careful. So I think they've been told that the last thing to happen will be the borders opening up again. So think 2022, not Christmas time. That's my. But then again, you've got to finish your degrees. Come on, plenty of time.
We have another question, if you've still got a few more moments.
Absolutely. Yeah.
About whether there are any sources of whether anything synthesizes Karrikins, because you said they were abiotic. Is that exclusive?
Well, you can never exclude all possibilities, right? So if it was going to be made anywhere, it would be microbiologically, would be my guess. So we have spent quite a long time, because we think Chi2 is a hormone receptor for something that plants make. We thought, well, let's look for Karrikins in plants. That was definitely we drew a blank there, right? So plants don't make Karrikins. Some bacteria make plant hormones. So bacteria are known for making cytokinin analogs, auxins, because there's a very close relationship between bacteria in the soil, basically the rhizosphere, and plant roots and plant leaves, and of course plants are infected by bacteria, right? So bacteria have a record of making plant hormones and analogs of plant hormones. So if it's going to be made by anything, it'd be by bacteria. And there are butenolide compounds made by bacteria as secondary metabolites that are they are antimicrobials. or they're regulators of biofilm formation. So the butynylide bit itself is certainly not rare. The other bit of the Karrikin molecule, what's called the pyran ring, after fire, is maybe less. It's less clear how that would be made enzymatically. But yeah, so we do not know. As far as we know at the moment, it hasn't been found biotically. But I would never rule anything out for sure.
So do you think the fire response is something that's been co-opted to a similar butynylide structure later in plant evolution?
So some plants obviously had an advantage from being able to perceive these Karrikins. And so the story that I didn't have time to tell, which we published earlier this year, is this weed called Brassica tonofortii. I've mentioned it as being very Karrikin responsive. And it doesn't require fire to germinate, but it's very strongly, it likes it, okay? And so it can invade after a fire, because it's one of the reasons it can invade is that it's so Karrikin responsive. And so, and what's happened in that species is there's been a genome triplication, and there are now 2 functional Chi2 proteins, and one of them is very sensitive. It's very sensitive to CAR1. So CAR1 is the Karrikin that is most abundant after fire. And it seems that Brassica has accumulated a couple of mutations. So we looked at the sequence of the protein, and we showed that there's actually one mutation that's critical that changes the protein into something that really likes CAR1. And so like we therefore infer and tell the story that that species has specifically adapted to responding to fire. And yes, so all plants have got this equipment, if you like, and some of them have also managed to use it to regulate their germination in response to Karrikins. And the really cool thing is, sorry, if I may just say, so parasitic seeds that respond to Strigalactones, they actually use Chi2 proteins that have evolved not to like Karrikins, but to like Strigalactones, because those are the proteins that regulate seed germination.
So would that suggest that fire responses are really very basal in land plant evolution?
Well, okay, so so you can do like phylogeography and try and work out when fire was really. So there was like a fiery Cretaceous time when oxygen levels were higher, and there were gymnosperm forests all over the place, and that would give you so that's not old enough to explain for it to encompass all land plants, right? It's not something that happened right at the early land evolution, but there have been periods in time when fire would have been more common, and therefore you might expect this to have evolved in different independent groups. So if you treat mosses or Marcantia, for example, with Karrikins, they don't care. So they can't respond to Karrikins, and even though they've got the Chi2 proteins. And my personal suspicion, for various reasons, is that actually Karrikins, as they stand on their own, are not bioactive, but they go into the plant and become metabolized in some way, which then activates them. And it's entirely plausible, given that as you go through land plant evolution, the size of the genome increases as you go towards the angiosperms, that basically higher plants have got the gear, the secondary metabolism, to activate Karrikins, but lower plants haven't. So that would also help explain why it seems to be something specific to seed plants. So it's again, loads of assumptions there, unknowns.
So the solution sounds like lots of quite complicated mutant screens to find.
Yeah. So I have one student who's looking at, so there are a class of enzymes called cytochrome P450s that are very abundant in plants. So Arabidopsis has got a couple of hundred of them or something, and they're involved in all sorts of metabolic pathways. So I don't expect to get a response from this, but it's possible that he will find a cytochrome P450 mutant that fails to activate or respond to Karrikins properly. We have done extensive mutant screens for Karrikin insensitive mutants, and all we found is Chi1, which is this MAX2, and Chi2. So we got about 14 different alleles of Chi2 and nothing else, which kind of suggests that either there's a lot of redundancy in how Karrikins are metabolized, or it's not done enzymically,
or it's lethal very early on,
or it's so bad that it's never really lethal. And teasing that part's really hard. So the other thing you can try doing is activation tagging, where you try and turn genes on randomly, overexpress the genes, which hopefully isn't lethal, but could lead to a phenotype. And other people are doing that. There's too much work. Although when I hear they're doing it, I think, oh, that's a good idea. And that's the trouble with so many ideas and not enough of these things, right? Just no people. Not enough people to do the work. There you go. That's the world all over, I think.
So would anyone else like to unmute themselves and ask questions? I've had a few direct messages saying that people have had to leave, but thank you very much for the interesting talk.
Well, you're very welcome. I hope you can at least look at it again on the recording.
And is there anywhere where you'd recommend for anyone who would like to look more into this stuff, a particular review paper or anything?
Oh, my website. Well, look, so on there, there's not that I've updated it recently, but my publications are on there, and not that they are the gospel or anything, but they will also cite other things that people can read. Plenty to be read.
So if that's it for any other questions, then someone's asked me about the stomatal response stuff.
Yeah.
So is that related to abscisic acid as that pathway?
All right. So I haven't done the work on stomatal responses. The people who have, so there's two problems. Yeah, one is the stomata, I think, they might be hyposensitive to ABA. So I'm sure they did ABA based assays, but I couldn't tell you exactly what the response is. And the other problem is that the cuticle, the waxy layer on the surface of the leaf, is thinner. So they lose water directly through the surface of the leaf and through the stomata. But yeah, I can't remember exactly whether the role of how it interlinks with ABA. I'm sorry.
It seems like a huge diversity of mutant phenotypes, certainly.
Well, yeah. So a lot of the phenotypes are related to auxin, we think. So if you look at what you can do comparative transcriptomics, right? You can compare a wild seedling and a mutant seedling. Yeah. What genes are different? Some of the genes that are dramatically different are related to auxin signaling. And that long hypocotyl phenotype is consistent with a mutant that's overproducing or over-responding to auxins. So it's hard to know, and the same with the leaf phenotype, right? That long petiole and the narrow blade, that's an auxin-related phenotype as well. So so it's hard to know what the primary and secondary phenotypes are. And yeah, it will be a kind of, I mean, it's true with anything. You mess up one hormone pathway, it has all sorts of knock-on effects. And then when you look at the plant, you're looking at the summation of all of that. Auxin is definitely a key player. It's a player in everything. That's why I daren't work with auxin, because I feel like I can never understand it enough.
So do you think this is upstream of auxin potentially?
In some processes, yes. I mean, I wouldn't say that it's like controlling auxin, but it is. I like to think of it as kind of modulating or sitting like alongside auxin, so that it changes the. We don't yet know. We think it might affect how rapidly, and I say we, the field, not me, how rapidly auxin is transported. So if it affects the ability of the tissue to respond or transport auxin, then it's upstream in that sense, right? So it's kind of changing the capacity of the plant to respond to auxin, but it's not necessarily regulating the production of auxin or anything like that. Although it might well be doing that.
Tried inducibly knocking out the Cockatoo to see if it sort of to separate it from a early development phenotype.
Glad you asked these questions, William. Yes. All right. So not inducibly knocking it out, but inducibly switching it on. So what you do is, or what we did, is you have a Cockatoo mutant, and you introduce a transgene that only gets activated with a chemical, in this case dexamethasone, which is a steroid hormone. And so we made two versions of it. We made a version that activates the wild type normal protein, and then a version that activates the mutant non-functional protein, so that we had a control, so that we could rule out the effects of the process of adding the dexamethasone, for example, which could have changes on the transcriptome. So we made those plants and activated both types of protein, and then harvested RNA 4 hours later, and then sequenced the RNA, compared them, and it was depressing how few genes changed. In fact, the guy who did it for me said, I think there's something wrong. Like, the data looks beautiful. There's loads of reads, but only about 50 genes have changed. That can't be right. And I said, no, that can be right, because we think the primary effect of the signaling pathway is probably not transcriptional, but it could be affecting auxin and affecting auxin dynamics and that sort of thing. And so there are a few genes that do change first, and we've got a small list of candidates to chase. And that, with the whole point of doing that, as you say, was to just get away from this problem of comparing wild type and mutant with all the differences that it entails. And so now one of the genes is a cytochrome P450, interestingly enough. It gets turned on quickly and strongly by Karrikins or by Cockatoo. But when we looked at that mutant, it didn't have we didn't have an interesting phenotype. Like, it still had a Karrikin response. So we don't know what that's doing either. That's Hugh's job, and he's stuck in China. Anyway,
so it looks like I've managed to bore everyone sufficiently with my questions, because the numbers of people on the live stream have been slowly dwindling as I ask more technical questions.
Well, look, I'm impressed they stayed to the end of my talk. So that's good enough for me. But
I think that's probably everyone. I should probably let you go. Thank you for staying up to talk to us.
That's right. It's dinner time. I've got home to go to.
Thanks very much.
You're welcome. And